Hello, in this video we will understand about the Warburg's effect. Otto Warburg was a German biochemist and medical doctor and Nobel laureate. Nobel Prize in 1931. So in this way, how cancer cell will be formed or progress the metastasis related to the Warburg effect in this video we will understand. This is the cell structure. Basically, whenever the glucose will increase, so in this way, due to the cancerous cell, due to the damage of the cell, increase the glucose uptake through GLUT1 channel, and in this production of more ATP through anaerobic respiration as a glycolysis to produce more pyruvate and more lactate to acidic environment, while mitochondria no any function aerobic respiration by the using of oxygen and produce carbon dioxide. This is known as the aerobic respiration through mitochondria through electron transport chain to produce more ATP, and this will be functionless due to the gene mutation, and in this the mitochondria basically will be dysfunction. In this way, here is the bigger picture of the biochemistry of the Warburg effect. So increased glucose uptake, aerobic glycolysis will increase in the presence of oxygen. So the aerobic glycolysis and functional mitochondria dormancy. Glycolysis produces metabolic intermediates for growth and biosynthesis of nucleotides and protein plus lipids for membrane. In this way, the glycolysis produce more um, constructive mediators like protein synthesis. So here is the glucose uptake through GLUT1 channel increase. Glucose convert into the glucose 6-phosphate to produce ATP. By this fructose 6-phosphate to produce fructose 1,6 bisphosphate, and this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate will convert into the 1,3-bisphosphoglycerate to produce NADH, and in this way, basically, this is the biochemical pathway. We are not concerning more to understand, but this is ultimately produce pyruvate, and the journey of the glucose to convert pyruvate to produce more ATP, and this end product is a lactate rather than the pyruvate will enter into the mitochondria to produce ATP aerobically. So in the presence of oxygen, no any in the presence of oxygen, lactate produce even with for example, the lactate will produce during the absence of oxygen and lack of the oxygen. So in this, the damaged DNA will progress more to glycolysis to produce more important protein and fat for synthesis of the cell membrane and maintenance, for example, the sustainability of the cell. The mitochondria, the reactive oxygen species decrease, and ATP from mitochondria will decrease, will lead to apoptosis also inhibit because mitochondria is important for the apoptosis of the damaged cell. And in this way, the 3PG will enter into the 1 carbon cycle to serine, convert into the glycine trihydrofolate, basically produced from the HCY, and the glutathione pathway. In this way, the MET will convert to again cycle run to produce NADPH, nucleic acid, lipid synthesis, and redox control. In this way, important thing you should need to understand, uh, the damaged cell should be recycled, and apoptosis is important, but the lack of the function of mitochondria will not progress the apoptosis of the damaged cell and produce more constructive or basically synthesis process to repair the cell but not repair the DNA. Pentose phosphate pathway produce NADPH and nucleic acid synthesis. And in this way, important thing you should need to understand the glucose uptake, and in the presence of oxygen even, the aerobic glycolysis will run to produce the ATP rather than the using of pyruvate into the mitochondria to produce ATP. In this, the glucose is short to understand, convert into the pyruvate, and pyruvate convert into the lactic acid, and lactic acid will lead to acidic environment. This acidic environment due to the lactate, and this acidic environment is important for immunosuppression. So the immune cell will not function for the engulfing of the damaged cell. So in this way, the acidic tumor microenvironment will lead to promote the breakdown of extracellular matrix and aid in invasion and metastasis. It can suppress the immune cell activity and contributing to immune evasion, not invasion. And on the other hand, in this way, the escape from the immune system will lead to cell maintain even in the form of the damaged DNA. And in this way, important to note that the cancer cell not due to mitochondrial damage related to the Warburg effect. Mitochondrial damage basically gene mutation to more enzyme production for glycolysis, and the glycolysis intermediates of biosynthetic molecules for DNA and cell replication. And in this way, the damaged cell will not recycle, and rapid growth by more glucose uptake by alteration of genes. More glycolysis due to more glucose, and enzyme for glycolysis increase. Cancer cell require glucose more. So in this way, the Warburg effect means more glucose used aerobically in the presence of the oxygen, while produce lactate to lead to acidic environment, and immunosuppression will lead to progress the metastasis and the cancer.